Hello, I'm Isla Castano and I'm about to demonstrate negative staining. First of all, make sure that you have all your supplies. You'll have the nigrosin, you're going to have your inoculation loop, your candle, and your alcohol, 70% alcohol, in order to sterilize your inoculation loop. You'll also have your colony that you've been growing for a couple of days, and then 2 slides. So first of all, you're going to sterilize your inoculation loop. Make sure you're going to dip it into the alcohol for about 10 seconds. Make sure that the inoculation loop is standing straight up and that you don't flip it over to pour alcohol so it doesn't drip down your inoculation loop. After 10 seconds, place the alcohol to one side away from the flame, and you'll have your flame here, and you'll run it over through the flame a few times, both sides, and make sure it's sterilized. Let it cool off for a few seconds, and you've already placed one or two drops of the nigrosin on one slide. And you're going to take your petri dish with your colony. Make sure you open the lid just enough so you can get the inoculation loop in without contaminating the colony. So you'll gently scrape up some of the bacteria, and you're going to mix the bacteria with the nigrosin with your inoculation loop very gently, and set aside your inoculation loop. And then with the other clean slide at a 45-degree angle, you're going to spread the nigrosin with the bacteria along the slide to make a stain. Make sure that it's evenly spread out throughout the slide. You'll set it out for about 10 minutes or so until it's air dry. So the reason negative staining is used is because it gives a more detailed look at the bacteria's morphology. This also helps prevent any distortion of the bacteria's structure. Negative staining makes the outline of the cell structure highly visible, since nigrosin and the bacteria cells are both negatively charged. And so since they're both negatively charged, this allows them to repel each other. It prevents the cells from taking up the stain, and therefore making it more visible once you look under a microscope. And since I don't actually have a slide here, I'm going to show you a picture, and this is courtesy of our last lab that we did with negative staining. So you see this background is the nigrosin staining, and then the actual microbes show up in a white or light colored, so that way you can see the morphology and the structure of the bacteria more clearly.